So this is the last of the videos about the applications of biology, and this time we're going to focus on genetic modification. The syllabus statements that we're going to cover are these here in black. I've added this red syllabus statement here because clearly there's a connection between these syllabus statements. To start off with, let's look at this one, 5.12, and this is all about the use of restriction enzyme and ligase enzyme. Now, before you bounce into this work, it's important that you are familiar with enzymes in general, and you might want to pause the video here and look at sections on pages 6 to 12 of the textbook. Remember that the enzyme has got a very specific active site. That active site is complementary in terms of the shape of the substrate. Then we've got this idea of almost a lock and a key, and then what we've got is an enzyme substrate complex, and when that forms, there's something called induced fit that happens lower in the activation energy, which allows the reaction to proceed. And then you've got the enzyme here that can be used over again, and you've got the product. So this is how enzymes in general function. So we need to think about restriction enzyme and what the specific substrate is. And we need to think about ligase enzyme and again look at the specific substrate and how that functions. As well as looking back at enzymes, you might want to look at the work about DNA structure, because you will be required to appreciate the structure of DNA. And remember, we've got that double helical structure. The two strands are anti-parallel, held together by hydrogen bonds between complementary base pairs. Also, it's probably a good idea to look back at the idea of protein synthesis, because what we're doing in all of these syllabus statements is we're working with different types of protein, and again the page references are there for you. So restriction enzyme, first of all, is an enzyme that is involved in cleaving DNA. That means it cuts up DNA at specific sites, and that allows you to cut out and splice out certain genes, and very often that's associated with the production of so-called sticky ends, and sticky ends are unpaired bases, which you can see in this next slide. So here we've got the DNA. Here we've got, remember, the complementary base pairs. Adenine always binds with thymine, guanine always binds with cytosine. Now, restriction enzyme is going to attach itself to specifically this particular base sequence, and then it's going to cut the DNA here, and then it's going to cut again at this end. So what we end up with are two pieces of DNA with these unpaired bases, and the unpaired bases here are referred to as sticky ends. And we use restriction enzyme as one example to cut out from a chromosome the human insulin gene. So remember, the human insulin gene is found on a chromosome, but we're just interested in cutting out a particular base sequence, so we can do that using this particular enzyme. And again, if you look at page 290 and figure 22.3, the textbook, that shows you a different diagrammatic version of this idea. So we've got to remember that we've got a eukaryotic cell, it's a human cell. Eukaryotic cells have a nuclear membrane, and then we've got these structures called chromosomes, which are made of DNA and protein, actually. So we're going to take out, first of all, the DNA, and then we're going to cut the DNA so that we can extract, in particular, just one gene, and in this case, it's the insulin gene. So restriction enzyme is used to cut out the human insulin gene. Now, why do we want to have sticky ends? So it's very helpful to have sticky ends, because if you've got the same restriction enzyme cutting two different versions of DNA, then what you can do is have the same sticky ends, which can then be annealed. So what we can see here is the action of this second enzyme called ligase, and ligase allows the hydrogen bonds to form between the sticky ends. But it's important to underscore that in order for this to take place, we have to have the same restriction enzyme that cut out this green piece of DNA as the same restriction enzyme that cut out this red piece of DNA. And the red piece of DNA here is meant to represent a plasmid. Now let's think about plasmids. You met them again earlier in the course, and this is a bacterial cell. A bacterial cell, remember, is a prokaryotic cell. It doesn't have a true nucleus. Its DNA is found in this nucleoid region. So here we've got the nucleoid region, and then we've got these very small circular pieces of DNA also within the bacteria, And those small circular pieces are the plasmids. These are very easy to move between bacteria. So one bacteria can take one plasmid and put it into a second bacteria, and that's how bacteria essentially do sexual reproduction. So what we want to do is to take out the plasmid from the bacteria, and we're going to use that plasmid in this process. And the next diagram is going to show you what's going on. So we've got the human gene here. We're going to take out the DNA from the human gene. We're going to splice out specifically the human insulin gene using a restriction enzyme. We're also going to take a prokaryotic cell. We're going to remove the plasmid from the prokaryotic cell, and we're going to cut out with the same restriction enzyme to create these sticky ends here. Then what we're going to do is we're going to insert the human DNA, the human insulin gene, into this section here, because the sticky ends are complementary, to produce what's referred to as recombinant DNA. And recombinant DNA is now a modified plasmid that we can put back into the bacterium. So this bacterium is then referred to as a transgenic bacterium or a transformed bacterium. And this particular bacterium is going to contain a piece of DNA which actually comes from the human. It's the human insulin gene. So this bacteria will express the human insulin gene, and it will start to produce insulin. It doesn't need the insulin, and so it will it'll take that, it'll do exocytosis and put the human insulin outside of its cell. That makes it easy to collect. Now, we don't want one of these bacteriums doing this production of human insulin. We want many of them. So what we're going to do is to take this particular bacterium, give it the conditions that it's required for it to do asexual reproduction, and we're going to produce lots and lots of these recombinant bacteria. So we take this transformed bacteria, asexual reproduction, so we've got loads of them, all of which are expressing the human insulin gene. So we're going to talk about, go back to the idea of transcription and translation, the production of the protein, which in this case is insulin, and the insulin will be thrown outside of that particular recombinant bacteria. This next slide is showing you very much the same thing. It's talking about, in particular, a restriction enzyme called EcoR1. And EcoR1 is a very commonly used restriction enzyme, and you can see it here in green, and you can see that it's got a very specific base sequence. This is the target site, so the substrate is the DNA, and specifically that DNA will fit into the active site of EcoR1 to produce these sticky ends. There are some restriction enzymes that don't produce sticky ends. Instead, they just cleave it straight down the middle. But your course asks you to think about these sticky ends. We've got ligase enzymes then allowing you to reanneal and to piece those pieces of DNA together. This again is a reminder. We got the human cell, the insulin gene, we got the bacteria, the plasmid, cutting it, recombinant DNA, transgenic bacteria, growing in culture, so we got lots and lots and lots of them, and then we can extract the insulin. Some words to just be careful with. When we talk about recombination, we're talking about recombinant DNA. It's not recombinant bacteria. Rather than recombinant bacteria, we say that we've got transformed, transgenic, or even genetically modified bacteria. What we've got here is a bioreactor. We met this idea previously, but what we're going to put in this bioreactor is the transformed bacteria with the human insulin gene, and we're going to make sure that we've got the perfect conditions for those bacteria to live and thrive and produce the human insulin gene. And the mixed product which comes out here is going to then be purified to produce that human insulin.